of California, Davis. Now, the title of his talk is Sorting of Polar Filaments by Multiple Motor Action. Now, please.
Thank you very much. It's a great pleasure to be here, and I'd like to start with. Okay, I'd like to start with thanking the organizers for inviting me. And I'll talk about a slightly different project than that advertised in the abstract book. I'll tell you a story about mitotic spindle, which is a fascinating molecular machine that segregates chromosomes in the cell before the cell actually physically divides. And the purpose of the talk is basically to illustrate how theoretical thinking about this machine complements experimental research and helps us to understand this machine. Here are the people who participated in this work. The main performer was my student Roy Wallman, who is shown here eating this cookie on which the spindle shape is actually painted. If you're interested in knowing more, I have much more information about some papers on my website. So before I get to the spindle, I want to introduce the main molecular player in the spindle, there are two types of players. One is microtubule, which is a long linear polymer, very dynamic, and the dynamics of it I'll show at the next slide. Here I wanted to emphasize that this is sort of a hollow tube. And it has very different minus M and plus M. So structurally, it's a polar structure. I didn't show characteristic sizes here, but basically the radius is a few nanometers, and it grows to microns, sometimes to tens of microns. And the second type of player is the so-called molecular model. Those are very interesting molecules which are able to burn chemical energy in the cells and convert this chemical energy into mechanical movement at work. So there are, largely speaking, two big families of those motors. Some of the motors called dyneins, and they normally walk from plus M to minus M of the microtubule. And another big family is kinesins that walk from minus and plus M. Though there's one renegade kinesin motor which also works from plus to minus M. And so one of the principal purpose of those molecules in the cell is to transport cargo, which can be anything. It could be other proteins, it could be big complexes, could be vesicle, could be lipid vesicles, basically packed with stuff. And uninhibited, those motors just move at a certain rate, which is normally of the order of micron per second. However, if one tries to stop them, they resist and try to continue moving, so they can actually generate forces if encounter some obstacles. So here's one of the most ubiquitous examples of how the cell uses this microtubule motor system. For example, in nerve cells, microtubules is basically a railway between the cell body with nucleus and many organelles to the axon terminal, where a nerve impulse propagates from one axon to the next one. And on this railway, molecular motors, which are basically locomotives, trains, they transport cargo from the cell body to the axon terminal and transport detritus, garbage, basically, like garbage trucks back into the cell body where it's recycled and so on. So this railway can be from 10 micron to actually up to tens of centimeters long. And they're very dynamic, unlike real railways. So basically the tracks constantly put down and dismantled and repaired and so on. So this slide shows you what is the most interesting example of the dynamics associated with microtubules. There's this process called dynamic instability, which is intensively studied in the last 20 years. And what it basically is, minus sense of microtubules is kind of static. Nothing interesting happens there. But the plus end cycles through repeated periods of growth, then shrinking, and then growth again. So there are sort of 4 types of processes. There's assembly and growth of this linear polymer, then event called catastrophe, then rapid shrinking, then event called rescue, and growth again, and the time periods between catastrophe and rescue are random, so there's this chaotic oscillation in the microtubule length. I'm not going into molecular details of what's going on behind this growth, catastrophe, rescue, shrinking, and so on. But it is connected to the process of hydrolysis, which is coupled to conformations of the building blocks that make microtubule. So here are 2 interesting examples, again from the cell life, which show how this dynamic instability is used in cell life. One is, this is sort of schematic outline of a cell which crawls on surfaces. So this is its nucleus, and this is the so-called lamellipod. It's the leading edge, which is engine driving the cell forward. Now, what microtubules do, they tell the cell where to go. So this is the so-called microtubule aster. It is focused in the microtubule organizing center, or centrosome, which is very complex but very small organelle, and all minus ends of microtubules are gathered, focused, basically anchored in this centrosome. And the plus ends are spread on the cell periphery. And so microtubules shrink and grow all the time, basically trying to sense what's going on at the cell periphery. And if the centrosome is in front of the nucleus, that's basically where the cell is going to grow. Because this microtubules will organize other kind of cytoskeleton at the leading edge, making it move this way. However, I will talk not about this example, but about this one. Here, dividing cell is shown schematically, and you see that instead of 1 centrosome, it has 2. And so those 2 centrosomes organized 2 microtubule asters. And both asters are engaged in interactions which make this bipolar spindle. So this is mitotic spindle I want to tell you about. And in next slides, I'll show you the spindle in much greater detail. So in our lab, we do both experimental and theoretical research on Drosophila. No doubt all of you heard about this remarkable organism, which is the cornerstone of genetics and basically of biology in general. And so we look at the early embryo of Drosophila, which is this remarkable multinucleated egg. So there, everything starts with 1 nucleus, and then it divides in 2, then in 4, then in 8, and so on, until there are tens of thousands of nuclei, and those nuclei are not separated by membrane into separate cells. So this is basically one giant cell with many nuclei. So what is good for our purposes, at some point, all these nuclei, which are originally all over the egg, they migrate to the surface and stay near the surface of the egg. And then it's great for light microscopy, because just with one field of vision, you can see hundreds of nuclei dividing synchronously. And that's what you saw in this movie. Here, microtubules are fluorescent, and you see, for example, if you look at this one, you see the nucleus, 2 centrosomes at the side of the nucleus, and the nucleus miraculously disappear, and microtubules extend into the former space of the nucleus, right, and make the spindle, and then something going on, and in the end there are 2 spindles, two nuclei instead of 1. So what's shown in this movie is basically schematically illustrated here. You start with 1 nucleus, 2 centrosomes, basically sitting on the top of the nucleus very close to each other, and you see this nascent microtubules. and then centrosomes start to migrate around the nucleus, and at some point nuclear envelope breaks down, and chromosomes confined in the nucleus, which are the storage of genetic information, come out. Microtubules grab them, segregate the sister chromatids into two separate parts, and then nuclear envelopes reform around the sister chromatids. And so you basically have the situation when you have two nuclei instead of one. And then everything repeats again and again. So our focus is trying to understand in detail this molecular machine. And gradually in this talk, I will show you what unimaginable complexity there is behind this simple scheme. And if we want to understand biology to the point when we start healing diseases, we have to understand every little detail. And the purpose of the talk is to show how difficult it is and what are the ways to go around this complexity. So I want to start with sub-project, which explains only one small stage of the mitosis. Basically, to start with explaining one simple thing, so that on this example you appreciate the role of theory, the role of physics in understanding biology. So what I want to show you is the stage which is called search and capture, which is very beautiful part of the process between this stage and that stage. So what happens when nuclear envelope breaks down? How come sister chromatids become attached to the microtubules? That's what I want to explain to you. So if you look at those two pictures, they kind of show the beginning and end of this search and capture process. So originally, as I said, there's nuclear envelope around chromosomes. And in the end, there's no nuclear envelope, and chromosomes become attached to the plus ends of microtubules emanating from two centrosomes. Now, another important thing is that microtubules attach not anywhere on the chromosomes, but there are organelles called kinetochores. So those are kind of tiny droplets of glue on the surface of chromosomes, so that if plus end of microtubule get in touch with this droplet of glue, this plus end is glued to it, so it stops doing this dynamic instability, it's fixed. That's basically shown here. You have one centrosome, in other words, sometimes it's called spindle pole, microtubule coming from it, so it grows, shrinks, grows, shrinks, grows, shrinks. When it bumps into the kinetochore, it gets glued to it, and this is called capture. So microtubules are kind of searching for this glue, and when they find it, they're glued to it. And when all the chromosomes are captured that way, then you have the end of this process. So the tricky thing here is, of course, the cell is not intelligent. It doesn't have eyes. It doesn't think. The microtubules are searching randomly in space. So they grow in random direction. And just by chance, they bump into the kinetochores and get glued to them. So the question is, if this random search goes on, how long does it take? Does the cell have enough time to assemble the spindle this way? So at this page, I don't want to overload you with mathematics, though mathematics is very simple. But I wanted to explain to you very simple reasoning, which was done by Foley and Leiber more than 10 years ago. And their corresponding paper, which was published in this year, made a huge impact on cell biology, because it was one of the first examples when physics was used very successfully to help to understand biology. So what they asked was the following. What are the principles of this random search that the cell has to follow to assemble spindle in the fastest possible way? How do you optimize the search? How do you make it fast? And the following is a very simple thing which you don't need mathematics to understand. So imagine that my ascent of microtubules is here. It's my my shoulder, and my fingers are the plus end microtubule, and I'm searching for kinetochore, which is this lamp. Right? So I don't know where the lamp is. I don't look at it. I just extend plus end here, there, here, do it many times until just by chance I pump into the lamp, and that's the end. Okay. So what should I do? First principle is I shouldn't have any rescues. What does it mean? If I grow microtubule this way, it catastrophes, starts shrinking, I shouldn't rescue it and continue growing. Because the thing is, the cell cannot turn microtubule like that. It can only grow it in one way. So it can start searching in a different direction only if you have new microtubule. So you shrink it to 0 and then start searching in the next direction. So if you have rescues, right, and if you start searching in the wrong direction, and if you have rescues, you waste too much time just doing this in the wrong direction because the chromosome is here. So no rescues. Another principle, you shouldn't catastrophe too fast. Why? Because let's say I'm searching in the right direction, and then catastrophe before I get to the target, right? And this is a shame because I look in the right direction, but I don't have enough patience, enough perseverance. I don't wait until I achieve my goal. So I shouldn't do that. I should grow for long enough time. The third principle, I shouldn't, on the other hand, grow microtubule for too long a time. Because let's say I'm searching in the wrong direction, right? Grow here. Then if I search for a long time, so I grow and grow and grow and grow, wasting time. And I grow in the wrong direction. So 3 things. No rescues, don't search for too long time with one microtubule, and don't search for too short time. So then when you translate those principles into mathematical equations, it is relatively easy to find very intuitive answer. You should basically grow microtubule to the average distance, which is exactly the distance between the centrosome and the kinetochore. As simple as that, nothing else. So with all this calculation, there's this simple answer. And then if you substitute the numbers which you can measure in the cell, you find that the optimal time, that if you do it in the optimal way, then the cell will achieve its goal in about 10 minutes. And the experiments, remarkably, are saying that indeed cell need about 10 minutes to assemble the spindle. And that was the great success of this theory, and it helped cell biology a lot. So our work was the following. The problem with this simple calculation in 1994 was that it was an estimate of just the process of one microtubule searching for one chromosome. That's why mathematically it was so simple. But the problem is the cell has, depending on which cell it is, but many cells have tens of chromosomes and hundreds of microtubules. And so then the question is, how fast can the spindle assemble? So there are 2 very simple qualitative things. So the problem with having many chromosomes is that really you have to wait until the last capture. So let's say you have 10 chromosomes, and the first one is captured in 1 minute. But it doesn't do you any good, because you have to wait until the last number 10 chromosome is captured. And because all these searches are very random, the more chromosomes you have, the longer you have to wait. And having, on the other hand, many microtubules is good, because many of them are searching, and whichever gets to the chromosome first, it does it. It's like a war. You have a big platoon of soldiers who have to capture some hill. The first soldier who does it, great. So another big problem is geometric. Kinetochores and chromosomes are spread over large volume, which is former nucleus. So it is very hard now to optimize this search. Remember, you optimize it so that you grow the microtubule on average from the pole to the target. But now targets are spread. So, the microtubules don't know which target they're searching for. So they optimize to certain distance, right? So they get targets effectively at this distance, but not at shorter distances, not at longer distances. So simple mathematics basically fails to deal with all this complexity. What you have to do is do computer simulation just directly simulating all the microtubules which are growing, shrinking in random direction, and getting the targets. And this is just a snapshot from this simulation. And the result was very unexpected for us. We found that for a characteristic number of microtubules between 250 and 1,000, the best we can do is get spindle assembled in hundreds of minutes, from 100 to 500 minutes. While the experiment says that really the cell does it in about 15-20 minutes. So that was the puzzle that we had to solve. So the solution, which wasn't known to us at the time, but which was just a hypothesis, which was basically unproved hypothesis, is that in fact the search might not be so random. So that we showed that with random search we cannot assemble spindle fast enough. So maybe it's not random. Maybe the cell, in certain sense, has eyes. So what are these eyes? Well, it's known in many areas of cell biology that the following thing can go on. You can have proteins which are switching between activated and inactivated state. Normally it just means that they attach either ATP or GTP, and they activate it, and then they burn this molecule and become deactivated, and they cycle. So if the burning process takes place anywhere in the cell, but the proteins are recharging on the surface of something, then you have the gradient of density of activated protein from this surface where they're recharging outward. And so the idea was that on the chromosomes, some molecule is sitting, it's most likely certain kinase or phosphatase, which recharges some hypothetical proteins. Later it was shown that this protein is called RAN, one of very important proteins in the cell. And so these proteins recharge, float away, get discharged, and this floating away and discharging creates this cloud of smell around the chromosomes. So chromosomes are kind of smelly. And the idea is that microtubules can smell them out. So what does it mean on molecular level? It means basically that, for example, the catastrophe is suppressed while microtubule is growing inside this cloud. And as far as microtubule gets outside the cloud, the catastrophe is very fast. So that's the idea. And so then we simulated on the computer this scenario, and this is the snapshot from that simulation. So say instead of completely random search when microtubules grow anywhere, in this scenario, microtubules only grow inside the former nuclear sphere. Inside the sphere, growth is also pretty random. But strangely enough, it gives order of magnitude improvement in the speed of the search. This just shows simulation of this RAN activated cloud. And so with this scenario, the improvement of order of magnitude, instead of 100 to 500 minutes, we have 11 to 48 minutes, which corresponds to observed 15-20 minutes. So that was our suggested solution. And then, fortunately, within a year from publishing our paper, it was first discovered that indeed there are a cloud of this protein called RAN around the chromosomes. So it's very likely that this RAN gradient does exist and it can bias microtubule asters. It was also shown experimentally that this bias takes place, though it's not completely clear if RAN suppresses catastrophe or something else. And second, there was very interesting prediction from the model. The model was saying that if you have a nucleus larger than, for example, 10 microns, for example, in oocytes, there are nuclei which are about 100 micron large. Then the model predicted that in such a large volume, even with a bias, there's no way to assemble the spindle fast enough. Okay? But experiment is that spindle is assembled fast. And so it was discovered in this paper that what happened in that case is the cell has kind of casts a fishnet around the chromosomes, which are scattered around huge volume. And then actin-myosin contraction, which is very similar to what's happening in the muscle, though sort of in a much more disordered way. Contracts this fishnet, gathering chromosomes closer to this microtubule aster, and only then microtubules successfully hunt for the chromosome. So this small project shows the value of physics and of mathematics and of these theoretical models in addressing relatively simple biological problems. In the second part of the talk, I want to show you the different end of the spectrum. In terms of, I want to show you unimaginably complex process of spindle elongation. And I'll show you how can you use theory to unravel what's happening in complex situations rather than in simple ones as here. So before we get there, I already showed you the simple schematic view of spindle development and mitosis. Here's the more complex view, which shows the stages of how two centrosomes separate around the nucleus, called prophase. Then there's prometaphase. That's where search and capture takes place, which I just described. Then metaphase. All the chromosomes align at the equator between two poles. So it's like a globe, you can imagine, of chromosomes or equator. And then anaphase, in which glue between sister chromatids disappears and chromosomes are pulled apart. This, by the way, was on YouTube about a year ago, the snapshots from a movie which students from University of California shot in the backyard in the swimming pool, which is of the shape of the dividing cell. Unfortunately, they did it late at night. They were drinking beer, and the photos are very dark. So if you can see anything on these snapshots, two people were performing, pretending that they are spindle poles, and 6 people in the middle pretending that they are kinetochores on the chromosomes. And they use this plastic, colorful sticks as microtubules. So they were waving the sticks around like microtubules. Then these people in the middle were capturing the sticks and then pulling themselves outward. So in the end, this pole pulled 3 sister chromatids one way, this one one way, and then, of course, they couldn't actually physically divide this. So it is relatively easy to understand what's going on in some separate stages of the spindle development. For example, when 2 centrosomes separate around the nucleus, what does separate them? Well, those are the forces generated by molecular motors that I mentioned in the beginning of the talk. So such motors called dyneins, which I already mentioned, sitting on the surface of the cell. They're basically anchored at the rigid wall. And the motors are trying to move to minus ends of the microtubules, to the poles. But they cannot move because they are anchored to the rigid wall. So instead, they're actually pulling microtubule, reeling it into the wall, and together with the poles. So they are generating outward constant forces. However, there are microtubules which are overlapping between 2 poles. And between those microtubules are other motors which serve as brakes. And those motors are called NCD. And those are also trying to walk to minus ends of the microtubules, trying to pull the poles together. And it's the fight of these 2 forces, one outward and one inward, which create the balance of forces, and one can compute the corresponding rate of separation of the centrosomes and predict the arc distance between 2 separating centrosomes as a function of time. And then when the simple model predicts this distance, you could actually measure this distance using fluorescent proteins. And you see that there's excellent correspondence between the model and the prediction. But this can be done only in this simple stage. Here's the complexity which normally horrifies the people. So what is shown at this slide is the list of all motors which participate. And there are actually more than 10 motors. So you have to, and those are types of motors. There are tens of copies of each kind working at the same time, all doing something different from their comrades. So you have to keep in mind all this unimaginable complexity of motors, plus there are 4 different populations of microtubules. There are so-called astral microtubules, which come from the poles to the rigid walls of the cell and interacting with dyning motors there. Then there are chromosomal microtubules, which come from the poles to the chromosome arms, not to the kinetochores, which was the first part of the talk, but actually to chromosome arms, and interact with motors called chromakinesin on those arms. And then there are interpolar microtubules, which basically overlap at the spindle equator. And then finally, there are kinetochore microtubules, which I described in the first part of the talk, connecting poles to the kinetochores. So you can imagine that just memorizing all these motors and all these chromosomes, which is required from the students of mitosis, can take months. Now we have to understand it in quantitative details. So what does it actually mean to understand all this in quantitative details? So first of all, any theory we come up with about the spindle should be compared to some data. So what is this data? The simplest kind of data is we can measure the spindle length, simply the distance between 2 poles. And this is the characteristic way this distance grows. The discovery of this curve about 10 years ago was a big deal in cell biology. So what happens, this distance stays constant for a while and then grows, and that happens in pre-metaphase. And then in metaphase, where the chromosomes aligned at the equator, again, the distance is more or less constant. And then there's this final stage of anaphase when the spindle grows again. So these intermittent periods of growth and stagnation are very important biologically, because, for example, in metaphase, why does distance not grow? Because there's lots of incorrect attachments of microtubules to chromosomes, which I didn't mention, which are cause of many types of cancer and death. And the cell pauses trying to repair all these incorrect attachments. So it is very important that the cell does this sequence properly, in proper time, with proper speed. And that's the reason why there are so many motors. So each of them does different tasks, and somehow this complex dance arranges so that cell progresses in proper pace with proper speed. And what we want to understand is what mechanistically is going on at all these stages. So, of course, just this one piece of data would not be enough. But fortunately, what you can do, you can inject many kinds of drugs into the cell or make mutant cells and see what is different in terms of spindle length in those mutant and perturbed cells. And over the last 6 years, the lab accumulated many such results. So this black curve is what's happening in normal wild-type cell. Then you can inhibit one of the motors, one of the kinesis, number 5. And then if that happens, the spindle just collapses. But there are some mild perturbations, which only makes the spindle grow in the wrong way, but it grows somehow. And then the cell actually survives, though with some severe diseased state. So we have all this quantitative data. The question now, what can we do with this data? How can we use it? So this is sort of the program from the general bird view, sort of. You have this mechanical spindle system, which I just described to you. What I didn't mention is that it's a vast biochemical network that regulates this mechanical system. And what it actually does, this biochemical network is basically like a light switch. It switches motors on and off at proper time. The thing is that at the present, we can't even start thinking how this system works. Nobody knows. We know many reactions here, but we have no idea how the actual switch operates. So we are not going to even try to understand it. We just will say that switches that turn motors on and off do it at a certain time. We don't know which time and why. And then we know the phenotype. We know how the spindle grows in different scenarios. So the thing we try to do is called reverse engineering. It's a very popular concept these days. It means you look at the results, and looking at the results, you want to understand how molecular system works. The analogy is like you have a car, but you don't have a blueprint to a car. So let's say you're given a task. It's like, for example, you captured spy plane from a hostile country. You want to figure out how this plane works. You don't have the blueprint. So what you do, you try to break it part by part. You break one part, see what changes in behavior of the plane. Break another part, see what happens. So it's the same thing. Here we break small parts, something changes, and looking at these changes of behavior, we want to understand everything about this. It's called reverse engineering. So I will gloss over fast about next few slides, because time is getting tight, and there are so many mathematical details here that I'm just afraid to overload you. If you want to figure out the details, I'm happy to tell you sometime in the middle of the conference, or I can email you a manuscript if you want to. It's now submitted. So we describe every molecular motors with 4 parameters. One is the maximal force this motor can produce. Another is the maximal velocity with which the motor can move. And it's experimentally known that in between, at some intermediate force, the velocity is smaller than the maximum. So we have these 2 parameters. And then we know that the motor goes on and it goes off at times which we don't know. So each motor is characterized by these 4 numbers. Time goes off, time goes on, force and velocity, and we don't know any of them. And we have more than 10 motors, so we have more than 40 parameters which we don't know. But if we choose these parameters randomly, we just toss the die and decide here's the force, here's the velocity, here are the times. What do we predict? So when we choose those times, we then start writing force-velocity balances and just compute the movements and changes in the spindle. And we predict forces as functions of time and so on. And this part I will just gloss over, not going to show you. So it's relatively cumbersome mathematical equations, but there's actually nothing difficult about them. There's just too many of them, more than 50 equations with about 100 parameters. And we describe all these 4 populations of microtubules this way, and collect all the equations together. And so there are 3 differential equations actually describing the spindle length, the distance between sister chromatids, the overlap between microtubules at the equator, and tens of equations which are algebraic, which just give certain conditions here. And then we solve all these equations numerically. Fortunately, with modern computers, this is the easiest part. You just crank the system of equations, solve them, and get this. This is the main thing. So after a computer solves the model, it predicts certain length of spindle as a function of time. And, and we know what actual spindle behaves like. And then, this is the important part, we score the model, meaning that we measure the error, this distance between predicted and actual behavior of the spindle, and assign score to the error. So the bigger the error, we have the lower the score, and the higher the score, and a low score means good. Big score means bad. And then, of course, we cannot do anything of this by hand. So we, of course, can write all the equations, but we cannot physically go over all the parameters. So then we use what is called a genetic algorithm, and this is the link to many evolutionary talk in this conference. So evolution was very helpful in this area, actually letting us making us to teach computer to behave like actual biological evolving systems behave. What it really means is the following. So that's what we do. First, computer randomly assembles about 1 million models. Okay, so we have supercomputer, which takes about a week to go through to move us from the beginning to the end of this process. It took another 10 minutes, and then I'm done. So this 1 million models are simulated, and then we arrange the models in order of scores. So the low scores means good models, high scores mean bad models. Then we throw away 90% of bad models, leave only 10% best models. And then the models start to, so this is survival, right? We throw away bad models. And then the good models start to mate and mutate, meaning we make the computer to couple two models, and models exchange the parameters. And so it's like parents passing genes with certain randomness to their offspring. And then the models also mutate, so we shift parameters a little bit. And then there's all this mutation recombination of the models, and then we do it for 200 generations, so it's like a few centuries of evolution. And then we have great models which perfectly describe the spindle. Still there's some lingering problem. So there are some good models, and in fact the problem is that there are too many of them. In the end we had about 10,000 models which were terrific, which described everything we know about the spindle, the wild-type cells, the mutants, everything. However, of course, the trick is that many of those models are very similar to each other. So from biological point of view, they all are one model. And then it's very difficult computer program of clustering the models, showing which one models are the same, which ones are essentially different. And then after we do that, let me skip over this, we just found one group of the models which is surviving. So we actually do have the answer how the spindle works. And over the couple of minutes, I'll just show you how it works. Unfortunately, it's not a big lesson for you guys because you don't work on mitosis. But for people who work on mitosis, it's what you need. So the computer predicts that everything starts with dynein motors pulling the poles apart, like I explained in the middle of the talk. And there are also chromakinesins on chromosome arms, which also push poles apart this way. And there are certain motors called kinesin 13 here, which eat minus ends of microtubules, trying to pull the chromosomes toward the pole and creating inward force. Then that goes on for a while, and the spindle doesn't change its length in this stage because those outward forces are balanced by inward forces. But then the balance is tricked by another kind of motor called kinesin 5, which pushes apart the interpolar microtubules, adding outward force, which breaks the balance, making the spindle elongate. And I'm not going to explain the next stages because it just overloads your brains. It's just what the computer predicts. Which motors go off, we saw that dynein went off, which motors come on, and so each motor that goes on and off just shifts the balance and make the spindle elongate as observed. So let me skip over that. So besides the specific times on and off, this model made very important predictions. For example, it predicted all the forces and velocities, which we didn't know before. And after this work was done, we actually did experimental study where we measured forces and velocity of two motors. And the predictions are actually confirmed. The measurements confirm the model predictions. Let me skip over that. And so this is what I want to finish with. So I just wanted to recapitulate our general approach to molecular systems and actually cellular systems. here's how we see it. You start with biological problem. First, you need quantitative data, and this is how biology of the 21st century is different from biology of the 20th century. In previous century, biology was qualitative. You could describe everything with words. Now it's not enough. You need quantitative data. For example, you need length as a function of time, just one of examples. So you need some functions which are coming from the experiments, numbers and functions. Then you have to form a hypothesis how the system works and translate this hypothesis, mechanistic description. And also you need actually multiple hypotheses. You need many, in all possible descriptions you can come up with. You translate all of them in mathematical equations, you solve the equations, and then the usual outcomes, you have too many possible answers. So then you need to do computer tricks. You have to do stochastic optimization, clustering, unsupervised learning, basically to weed out all the bad solutions and to cluster the solutions into biological scenario. And then you have to think, what does it all mean in biological term, completing the circle. And this can be done for any kind of system. For example, instead of mitosis, you can think about cell migration, do the same. Start with the program, get some quantitative data, come with qualitative scenario, translate it into math, solve the equations, do computer tricks, go back to biology. And the big future challenge, which we have no idea yet how to attack, is, for example, add biochemical regulation to this mechanical model. And the problem is that, according to our estimates, even modern supercomputers are not enough to deal with that. So we estimated that few years of simulations of supercomputers, for which, of course, nobody has neither money nor patience, would be needed to sort out, to apply this paradigm to this problem. So this is sort of open program for now. And I wanted to end with the following. Just tell you about some very general thoughts about design of complex systems that come out of this thing. First, we understood that what the cell does is use the two principles which are called robustness and redundancy. So the spindle turned out to be very robust. This is experimental fact. You can hit it with many kinds of drugs, assault the cell with many mutations. It still goes through mitosis, worse and worse, but it still works, it still survives. Until, of course, you hit it with so many perturbations that then it just dies. One of the reasons for this robustness is redundancy. Because, as you saw, there's this overwhelming variety of motors. Why there are so many? We actually found a scenario for which just 3 motors can get the spindle from the beginning to the end. But then the spindle is not robust because you have just one mechanism. You injure this, kill this one mechanism, you kill the cell. Well, if you have 10 motors, they're of the order of 25 different mechanisms of elongating the spindle, and they are redundant. So you can injure one process after another, still spindle survives. Then one thing which is actually easy to appreciate, the cell in all processes it does is an open system consuming lots of energy. If you estimate how much energy a cell consumes to the spindle elongation, and then compute its effectiveness, how much of this energy is actually converted into force and movements, the stunning answer that the cell uses only 0.1% of energy well. The rest is wasted. And the interesting question is, why is the cell so wasteful? Because it's hard to feed our cells we have to eat constantly to maintain this stuff, and most of the energy we lose. Well, the answer is that this is to be so robust and redundant. So the cell has to do lots of things in vain, but it does it faithfully and precisely. Another point is the cell always solves optimization problems. And the interesting thing is that it does what is called multi-objective optimization. For example, the spindle has to be assembled fast, but also accurately. So what is the emerging theme in mitosis? is that the cell finds the optimal compromise. If it does it too fast, too many errors, too big chance of cancer. If it does it very accurately, then it's not fast enough. You can die because you don't procreate fast enough. And finally, I noticed from the program of the conference that it will be lots of very philosophical and Buddhist connotations here. So I just wanted to emphasize two of the noble Buddhism truths. So you can see from all these molecular machines, certainly you can see interconnectedness. All of these processes are connected to everything else. And impermanence. So all of these machines are there, alive and functioning for a very short time after that they are gone, which is no tragedy because it's all part of the whole. And on this I wanted to end. I just wanted to comment that this is a fragment from Diego Rivera's mural. And Diego Rivera was actually obsessed with mitosis. It's one of the unknowns fact of his biography. So what you see here is the calloused hand of a big proletarian worker, and it keeps the crystal ball in which you have the cell undergoing mitosis and those clocks. So in my mind at least, the science of mitosis was always connected to art, which is one other interesting part of Buddhism. Thank you.
Thank you very much for an interesting talk. Now, here.
Cerrito, University of California, Santa Cruz. I'm presenting here. I'm doing interdisciplinary philosophy. The question is from philosopher. We have very interdisciplinary symposium, conference here. And everyone has idea what is life. And this is my suggestion. If every participant in the end of his speech would say couple of words, just couple of sentences, what is life on his point of view? So this is my direct question, what is life on your point of view? Thank you.
I'm in a very enviable position to just say I don't know. Because I'm handling molecular machines, which are, you saw how complex they are, but they are nothing compared to brain and human beings. So I pretty much have an idea what molecular machine is. And molecular machines are certainly alive. So my definition would be maybe, it's a very complex, heterogeneous, diverse, reproductive open system consuming lots of energy. But, this is, of course, it just scratches the surface of what is life. I don't know what life as a whole is.
Sergey here, my colleague, spent a Fulbright year in my lab, and he has a poster I'll just call your attention to. And he, at least in the poster, says what life isn't, and tries to constrain what life is in that way. So I recommend dropping by Sergey's poster. Often enough, biologists go to ever simpler systems to try to understand the sorts of amazing complexity that we see in this eukaryotic cell, fairly advanced organism. Are there simpler versions of mitosis that we can learn something about the evolution toward this complexity that you have now put together?
This is actually a terrific question. We studied Drosophila spindle, which is one of the complex ones, for the reason that genetics of Drosophila is studied to death. So there's a lot of experimental tools attacking it. There are simpler spindles. One favorite spindle is yeast spindle, which is order of magnitude simpler. And it's also vastly studied. However, there are even simpler systems. For example, what is very fashionable these days is the so-called reconstitution in vitro biology. So people actually break the cell into million little pieces, pick up just 3 out of 100 proteins, put them onto lipid vesicle, and resurrect the spindle and see how this resurrected spindle works. So this, I think, is probably the most promising way to get the spindle. And finally, it was this view for the last century that prokaryotes don't have proper mitotic spindles like I showed here for eukaryotic cells. Well, fascinatingly, in the last 10 years, some kind of primitive molecular motors and dynamic polymers were discovered in bacteria. And this is one of the hottest subfields of cell biology, so those also would be very good to understand. But it's, again, it's an excellent point. The point is to try to find the simplest possible system which gives the understanding.
Okay. Yeah.
I have a comment and a question. The comment is, I find it curious that you showed Darwin's picture in the cartoon at the end where you illustrated your schema. And you put the picture in the place where you had a genetic algorithm entering the scheme. Now, it seems to me that the essential feature of a genetic algorithm is to generate variation. And that, of course, was the one thing that Darwin did not know about in his theory of evolution. So that's just a comment. The question is the following. In your model, what guarantees that sister chromatids go to opposite poles? In other words, how do you get correct Mendelian segregation?
That's also a good question. And I mean, we have the answer. The answer is that first, the kinetochores have to be captured properly, meaning one of the sister chromatids is captured from one pole, another from another pole. Another is self-assures that there are always outward forces trying to pull the chromatids apart. If you have these two things, then you're successful, because at some point the glue between sister chromatids dissolved and they're pulled apart to separate poles. Now, how the cell designed those 2 or 3 principles? It's a big question. We don't know. What this computational theory does, it gives us tens of scenarios how it can be done, and cell chooses one of them. And I think it just happened in the course of evolution. So the cell tries different things, and after trial and error, got the right one.
Okay. Yeah.
Have you come into contact with the possibility of the electromagnetic fields being involved in this very special system with these storms of things passing out and in? Did you ever see any comments on that?
No, it's a very good question. We try to completely stay away from that part. And here's the reason. This always happening in cell cytoplasm, which is a very strong polyelectrolyte. And the problem is that there are neither experimental nor theoretical good understanding of what this complex polyelectrolytes of this sort, how they behave and what are the principles. It's clear from some indirect, there are some indirect experimental hints that electromagnetic part is very important here. All these motors, all these polymers are highly charged. There's lots of counter-ions floating in the cytoplasm. But we are far, far from the stage where we can do anything about it. Too complex.
I'm happy if you try. I'm Leif Salford from Lund University in Sweden, I forgot to say. Thank you very much.
Okay. Yeah.
You can use mine.
Okay. Thank you. My name is Matsui, Kyoto University. I am an environmentalist. So that's the, I did not know about the very precise mechanism of spindle. But I now understand, which is very interesting, of course. And then my question goes to the interaction between environmental contaminants and in the mechanism, especially the nanoparticulate. Because, just a moment, nowadays, nanotechnologies are really rapidly expanding and producing variety of the nanoparticulates, inorganic or organic. And because of the size of a nanoparticulate, the electrophysical or electrochemical properties are really different from the normal large size particle, nor the molecules in solute. So that's, when I see your very precise mechanism of the microtube and conveyors, 2 conveyors, one go to the minus and the other one go to the plus. So those nanoparticulates really electrophysical and chemical properties. And I'm very much afraid of those interference of those chemicals in this process.
Thank you. It's also an excellent question. The problem is that in this case I have to plead complete ignorance. We are doing only fundamental biology, and I know very, very little about environmental pollutants and effect on the spindle. One thing which we are very interested in is the types of cancer which are associated with wrong separation of the chromosome. And there are many types of conditions that cause this type of cancer. The thing is, I only know, so what we do is use certain very strong drugs to induce those conditions, to study this type of defects. And it is known that many other different subtle conditions, among them these nanoparticles, can cause this thing, but I know almost nothing about it.
Okay. I would like to have a final question.
Can I have a short?
Yeah.
So this is a short question. In your first part of the talk, you talked about the searching of problem. And then did you include elasticity of the filament in your simulation?
No, not really. It's also, you're asking this excellent, very smart question. So this is a very smart question. The thing is that due to elasticity of microtubules, they tremble.
So maybe it improves your result.
Right. It makes it better for the cell because besides this kind of search, they just do this lateral search a little bit, and it does improve things. But in the simple model, we didn't use this fact at all. The thing, of course, that model is terrifically oversimplified because besides this lateral search, there's so many other things I just didn't have time to talk about, which are of much more drastic consequence than this elastic thing. So I'm happy to tell you more about it sometime.
Okay. Now, thank you very much again.